This thing, I don't know what this wants me to do. It says it's working. Okay, whatever. All this. Oh my gosh, this thing. Whatever, I guess we won't mess with that. And this. You guys are getting signed in. I hope that you guys got the email saying that we are not going to be doing our assignments on Top Hat anymore. You can still use it as a reference. It's still your lab manual, but we will be doing instead the pre-lab questions on Canvas, which are easier, and so that we don't have to mess with Top Hat, guys. We don't have to mess with it anymore. But I just have had too many complaints about it not working for people. I mean, last with Top Hat, with how it's like laid out, or yeah.
It's really it's laid out really well. It's weird. So I would just kind of read the question and then go back over to the information, like what information I needed.
So I would say this, that like in the future, if we had gotten to exercise 2, which people just weren't having it, so that's fine. But exercise 2 and everything past that, all of the questions are just at the bottom. They're not like throughout it, but you don't have to worry about that now because we're not using it. So now you can just go on to Canvas and answer the questions that are on Canvas, and you can use your Top Hat Lab manual as reference if you want to, or you can use like those handouts that I told you guys about. They are on Canvas as well. So the ones that we turn in the back of it. So yeah, so it's going to be easier for your uploads. You guys haven't had to deal with the uploads, but this is why I'm bringing this up, is because all of the other classes before you were complaining about that and complaining about other things and whatever. I've used Top Hat before. The previous semesters, people didn't have as much problem with it, but I have found that people come in hordes. So it's like people that have an issue with it. It's like you don't just have like one class or a few people. It's like my whole semester will have a problem. And I don't know why it happens that way, but that is legitimately how things work. It's yeah. So some semesters you get students that have like certain mindsets just all in one semester. It's just weird, but that's how it goes. And this is a group that doesn't work well with Top Hat. I don't care for it anyways. So it's not a big deal. It just sucks that I can't give you your refunds back to you. But do try to use it for reference. It does have more explanation to it. And when we get to the pond water and stuff like that, I don't have like as many pictures in the handouts, right? So it has a lot more pictures to refer to for stuff like that. We'll need it for that when we do the pond stuff. So, but yeah. So I hope nobody's like too heartbroken about that. But now every single thing that you will need to turn in for this course will be on Canvas. So you don't have to worry about going to anything else. Anybody have any questions about anything before we move on to today's stuff? Okay. I had a student in my last class for you guys. She was saying that she heard from one of my other sections that you can't use anything except for the text for the, what is it called? The group projects. And that's not true. You can use whatever you want for reference. Like if you want to pull, images off of Google or like find things on Wikipedia and whatever and use that, it's fine. I just wouldn't try to put more information than what's in the text. That's what I'm saying. So yeah, you can use whatever you want to put in whatever, but just you don't need to put any more information than what is covered in what we learn in class. That's what I'm trying to say. So I hate for you guys to turn in, projects that you guys thought was really boring and that wasn't any fun to work on it or something, just because you thought you couldn't do something else. I do would appreciate if you guys would try to have some fun with these projects. It's meant to be lighthearted. This is meant to be easy points. It's meant to be not a super challenge for anybody. Yeah, I want you guys to work together, and yeah, I mean you have to cover your topic, but this isn't meant to be like a big test sort of a thing. So yeah, meant to be kind of straightforward. Anyway, that way. My husband came up with an idea that you're probably going to hate. Yeah, so we'll see how the Unit 1 projects go. Obviously not next week, but the week after that, right? You guys are going to do the presenting of them. No matter what format you guys turn in, you have to present it in front of the class. So just explain like what it is and a basic rundown of it. But we'll do that on the Tuesday before the exam, right? And then the exam's on the Thursday. And so without those projects, once we see how it goes with Unit 1, and then Unit 2, I'm hoping that you guys will enjoy it a little more because you'll have an idea better of what to expect. I've not done these projects before, but I think they'll be useful in helping people study. So I don't have any examples to give you guys. Unfortunately, you guys will probably be the examples for the next semesters. So think of that. But we'll see how Unit 1 goes. Unit 2, hopefully you guys have a little bit more fun and you feel a little bit more, I won't say competitive, but like you'll see how other groups maybe are a little bit more creative and you'll want to, look at it more that way rather than like a test in this big old whatever. Then Unit 3, when we do Unit 3, yeah, of course it'll be the same concept of whatever, but my husband suggested that we have you guys vote on who, and it's not many people in this class as there are in other classes, but have you guys vote on who you think, what group you thought did the best, maybe that semester or in that unit or whatever was your favorite group. It can be anonymous voting. We will probably do it like electronically or something after the third one, and then give a trophy to, based on that, right? So a trophy to first place. We might probably give something to everybody, of course, but yeah, look at it that way. So I think that would be fun. Yeah, give a little bit of more fun. It's not.
Nobody loses.
Nobody loses, exactly. And I don't know if I've said this to you guys, but people are so stressed about the questions, right? Like whenever I'm asking the questions in the class, I mean, you guys are picking up on it. That's not meant to be a stressful thing, really. But some people are just like, oh, if I'm not getting the right answer and the group's not getting the points, I'm like, you guys, I tell you pretty much that you're getting points, yeah? And it's really like the only reason that we have that competition going on the front page right now is we haven't just gotten enough of these polls to have them be equal. So don't worry, it'll probably work out just fine. And as long as everybody's trying and we're all engaging and we're trying to answer, even if it's wrong, you guys know you're probably gonna get your point anyway. And you're probably gonna everybody get full credit. I know that some group might get farther ahead, but as long as you're all working on it, you'll probably end up with full credit. But hey, you can look at it now and see as we get further into it, maybe that can be another motivation. Let's get our group more points than, Delta or something like that. So yeah, it's playful. It's right, not meant to be stressful. Yes. Absolutely fine. So exercise 1 is pre-lab. I've already entered the grades for them. Might be a couple that I missed because some people just for this course had not quite completed it before the lab, and that's fine. No worries. So if you don't have a grade in there for pre-lab 1, it's just because I haven't entered it in, not because you didn't do it or something. I have, so that's it. Almost everybody has done. I mean, I don't think anybody has. There's like one person that literally didn't do pre-lab 1 for all like 80 of my students. So no worries, you guys figured it out just fine. And that should be fine. We also have, or if you were one of the people that worked ahead and you did the pre-lab for 2 or 3 or 4, because I have some people that did that. your credit will carry over. If you were to decide to do the questions on Canvas, then, and you've got a lower score on it, I'll keep your score on that. So if you were somebody that worked ahead, you'll still get your full points. FYI. But yeah, everybody else will just switch to Canvas, and yeah, it'll be easier all around. All right, let's do this. True or false? Since bacteria don't have a nucleus, they can't contain DNA. Erase this on the board. Okay. So you guys did better than my other classes, honestly. My other classes had about 50-50. But so just to summarize, just because they don't have a nucleus doesn't mean they don't have DNA. Their genetic material will still be DNA, and it's just floating out in the cell. It's not contained by a membrane. That's really the difference there. Anytime you have a cell, and we remember how we categorize what cells were, right? We said it's the eukaryotes, the bacteria, and the archaea. They all qualify as having cells. They all have DNA as their genetic information. When we move into the viruses, the story's going to change. So, yeah. Cool. All right, the peptidoglycan layer is blank in gram-positive bacteria compared to the gram-negative bacteria. Okay, wonderful. So most of you guys getting this one. The gram-positive, they're positive for that thick peptidoglycan layer. So they have that thick layer, whereas the gram-negatives have the thin layer of peptidoglycan with the membrane on the outside. I think it kind of is going to ask us about the membrane. All right, gram-negative outer membrane contains which? Okay, so endotoxin is the correct choice. This is one that you might, yeah, if you don't know the bacteria as well as I do, probably have to revisit a note on at this point, but that's okay. I do want to point out something with this as well. The way this question is written. So we have kind of an obvious question, what's in the gram-negative outer membrane, is basically what it's asking, and then you have some choices. And they're all choices about stuff that have to do with stuff we've learned, or at least we will be learning about life design here today. But yeah, they're all science related or bacteria related. We know they can have exotoxins. That just means that they're making it and pushing it out of the cell. And lipotechoic acid is something that's in the gram-positive, but it's not a toxin, right? But anyways, these are all terms that kind of can be related back to what's going on in the gram-negative membrane. I'm putting toxin in there because I know that you probably might think, oh, it has a toxin in it, and then which one would it be? This is the kind of question that you will see on the exam as far as like what, I won't say the simple ones, because I will have some really basic ones, but like the lower level ones, okay? Most of the exams tend to be more like this kind of a question. There will be some more difficult ones on there though, so beware. Like there'll be the scenario ones, you know what I'm saying. Okay. What is acid-fast staining? All right, so acid-fast staining. This is staining for mycolic acid, which is the waxy substance that we see on mycobacterium, which I know the next question has to do with that type of a thing, so I'm not going to give away too much, but that mycolic acid, that waxy coating, And this acid-fast staining involves putting a stain that is lipid soluble on there so we can stain for that waxiness and see if it is there or not. And that's pretty much what it relies on. So, yeah. Okay, moving on to the next one. All right, true or false? So this is mycoplasma. Is an acid-fast positive organism with a thick, waxy cell wall. Is that true or false? All right, no judgment for me because we're still new at this. This is going to be, so mycoplasma and mycobacterium are the two mycos, okay? They're the only two mycos that we need to know for our course, as far as like genera, so mycoplasma and mycobacterium. Mycobacterium is the one that has the wax, the mycolic acid wax, whereas mycoplasma focus more on the plasma, right? We said pouring it out and it doesn't have a shape. And why it doesn't have a shape? The cells don't have a shape because they don't have a cell wall. So that's how we can kind of, I remember it anyways. Yeah. Reminded you of that amorphous sort of shape. Yeah. Some of my slides have stuff to do with what's going on. Some of them don't, but some of them are like that. We're sort of like cheeky. I did actually think that one through. So what are bacilli? I'll give you a little bit of a hint. Remember bacillus? I remember coccus. I don't know why this gives us so long to answer this one. Hurry, guys, you only got 98 seconds. All right, great. So these are the rod-shaped bacteria. So yeah, I don't know what else to say. And the cocci are the round ones, right? What does peritrichus mean? All right, excellent. So you guys got this one. You did really good at it, actually. So the flagella distributed all over the entire cell. This one, knowing the four arrangements is a really good one, because the questions will be something like this, where you might see, oh, what does peritrichus mean? I know that had to do with flagella. Hey, I can answer it now, but now you can see, knowing that has to do with flagella is probably not enough. So knowing the definitions of those four, try to make them as simple as you can on yourself. Okay. Bacterial capsules. What about them? All right. So majority wins with this one. This is going to be protecting against phagocytes. What was the other big? It really has two major functions that we're concerned with. Yeah, it's going to be evading the phagocytes or the immune cells and adhering. So it could be sticking to surfaces or creating biofilms, that sort of stuff. When are endospores produced? Okay, so endospores are going to be produced when, I mean, basically like when shit goes south, right? So if things are going bad for you, if it's rough times, let's say that, okay, rough times, you can't find enough food and, you know, it's feeling a bit almost apocalyptic for you, then you're going to hunker down basically and try to wait out the storm. That's what an endospore is. It's this really resistant, it is dormant, it's not doing anything, it's just waiting to get woken up again by more nutrients or something like that. So that's what the endospores are going to be doing for the bacteria. So they won't be produced, the endospores themselves won't be produced in these other situations because these are actually pretty good times situations. Yeah. You won't be replicating unless you're in a good time. All right, how would you describe the strepto arrangement? Okay. In case you haven't been picking up on what I'm writing up on that board, we're about to get real comparing to some endospores. So that's why I want you guys to be sure that you understand what an endospore is, because fungi and protozoa are going to be making structures that are going to make you even more confused. Okay. So chains, exactly right. You guys got that one. Strepto is like a strip. Okay. So let's look at our little podium real quick. Yeah. There you go. Yeah, it works. In little plumps, little, yeah, I don't know what to call them, little bumps, and yeah, that's a really good way to think about that. Yeah. Your throat is long, it'll go around the whole length of the throat, and then the skin having like, you just have like what boils or something like that in little clumps, so clusters or whatever. Yeah, that makes sense. I like that. To put it into practice actually. All right, so good job, guys. We did great on this. Let's move on to chapter 5. Chapter 5, we're going to talk about the eukaryotes, and that's going to include talking about, go away. It's going to include talking about mitosis, which I know that you guys just can't wait to hear about. All right, let's do it. I don't know why the mitosis is in this chapter and not in chapter 6, but it is what it is. We're going to talk a little bit about endosymbiosis as we go through this chapter. This is kind of introducing it a little bit early, I feel like, but when we get into the mitochondria and the chloroplasts, I'll really go into more detail on it. But it basically is the concept that there was a progenitor cell that swallowed up a bacterium, let's say, and then they both were benefiting from each other. The bacterium was making energy for the big cell was providing food to the bacteria, for example, and then that became a permanent relationship. And then that's where some of our organelles came from. So that's the endosymbiotic theory. It might sound wild. It might sound like, yeah, sure, our parts came from bacteria. They were once bacteria. Sure they were. How would you even know? But I'll show you how we would know, but not right now. Okay. All right. Here is the structures of the eukaryotic cell. Every single eukaryotic cell has everything on the left, and we're going to go through the parts. So no worries on having to worry about what they are right now. But here on the right, we have some things that when you actually take a minute to step back and look at it, makes sense that not all of them have these things. So cell wall. Our cells don't have a cell wall, right? We know that plant cells do. Our cells aren't rigid like that. Plant cells have a cell wall, and fungi have a cell wall, but many other cells don't. So not all of them have a cell wall. Locomotor appendages. These are things for moving around. Now, would our cells, for the most part, need locomotor appendages? Absolutely not, right? So obviously there are eukaryotic cells that don't need locomotor appendages, but obviously there are some that would. So we might not think as obviously about this one because we probably don't think often at all about our eukaryotic pathogens. People often think they're probably bacteria, and they're probably wrong. But we'll talk about them. Things like, what do I want to say? Giardia, which is something that can pollute the water and make you say pollute, but it's just a protozoa. and I can make you have really terrible diarrhea. They say like 2 months straight of greasy, malodorous stools. So that's what Giardia lamblia causes. And if you've ever heard of giardiasis, that's what that is. There's also like trichomonas vaginalis, which is a trick, which is a sexually transmitted disease. It causes vaginitis. Vaginitis is also caused by yeast infections, but here I'm talking about a sexually transmitted version. Trichomonas is a protozoa, and it does have flagella, and it can move around and stuff like that. You can see them quite clearly on microscopes whenever you do get samples of them. When you work at Southwest Integris, you see quite a lot of these because there are a lot of hookers coming in and getting tested. I am not kidding. So, all right. So anyways, and then we have the chloroplasts. Obviously, the chloroplasts aren't going to be in every cell, but we know plants have them. We know algae can have them, but we don't need them. So not all of them need that. Let's talk about these parts. Oh, here's a picture. This might be a good picture to come back to. It doesn't have the explanations on it, but that I think might be good, useful. I dare you guys to go back when you're studying and find one of these boxes and then explain to yourself what it is that does. Okay. All right, let's get into it. We talked about flagella in the bacteria. So why not talk about the flagella in the eukaryotes? Okay, so they are not the same. They help with locomotion, both of them do. But whenever we learned about our bacteria, remember on theirs, here's the little butt of your bacteria, right? And it's got its little motor that's going through its little, whatever, its membrane there, and cell wall if it has one, whatever. And then its little flagellum hanging out. It's like protein compounds just chilling, but it's out there open. And then how does it move around? You guys remember, right? This is going to rotate one way or the other, clockwise or counterclockwise. So that's the bacteria. The eukaryotes are much different. They'll have on the outside of their cell whatever, and then this is their cell outside, and that's their flagellum. Do we see the difference? See how that outside membrane is continuing to cover the outside of the whole thing? That doesn't happen with the bacteria. And then how are these guys going to move theirs? They can't move this whole length of theirs up and down and wiggle it and whip it. So it moves in a whip-like motion. So they have way more control over their flagella than the bacteria do. It's 10 times larger than the ones in the bacteria as well. And yeah, so it's a sheathed cylinder. So we've got our membranes around the outside of it, and inside we have microtubules. And these are just like protein-based structures inside that just give support. And they say it's a 9 and 2 arrangement. I'm going to show you what I mean by that. But yeah, so this guy can actually move like this. And then inside is the actual structure, the microtubules. They'll go all the way down, but you get the idea. They'll be going through it like that. So here is what a cross-section of a flagellum would look like for the eukaryotes. This is our outside membrane, this part right here. And then these guys that are little circles in here, these are our microtubules. So when we say 9 and 2, there's 9 of these pairs. And then in the middle, there's 2 just chilling in the center. Okay. So that's the 9-2 arrangement of how that's what supports the actual flagellum for the eukaryotic cells. Way different, but still going to allow for locomotion.
So
yes.
Like,
yeah.
Question, but like.
So a sperm. So yeah. Yes. Because because it is one cell,
right,
and it does have the ability to swim. So yeah, it's a flagellum. Yeah. Nobody's ever asked that question and maybe put it in that context, but you're absolutely right. No, you're absolutely right. I'm glad you're asking it. All right, so cilia. So with the eukaryotic cells, we can move with flagella, but bacteria can move with flagella. But that's all the bacteria get if they want to move. With eukaryotes, we have a few options. We have the flagella, we have the cilia, and there's one other thing that I'll come back to later on. But for now, we saw the flagella, this is the cilia. These are little hair-like appendages that kind of beat, usually in a liquid or whatever environment they're in. And we see these on the outside of some kinds of protozoa. They can use that to move around, or we can even have these in animal cells. You have cells with cilia on them. For example, in your lungs, you have cells in the epithelium of your lungs that have cilia on it to help move mucus out. So you'll make mucus, and it'll catch things whenever you breathe, and then the cilia help move it out, and then you cough to help expel it. And that's how that helps to keep your lungs clear. So that is a very important function in your lungs. And when you have something like whooping cough, which we'll learn about later, you cough so much that you no longer have the cilia, like they just get obliterated in your lungs. Yes.
So like for smokers, when you look at an X-ray and it's like all black or whatever, is the cilia dead or what is?
They probably still function, but they're probably not as successful as somebody who has healthy lungs otherwise. So that's a good question too. I don't know. I never actually looked at it like at the microscopic level myself, of course, but it might be worth looking into. Anyway, so yeah, cilia, important. If you didn't have any at all, like if they got obliterated by something like whooping cough, then you would be way more susceptible to things like pneumonia and other infections like that. So that can be a real concern. But yeah, let's move on. Glycocalyx. We talked about the glycocalyx in the bacteria as well. So we're back on that again. This was the one that where we had the slime layer and the capsule. And you'll notice that it is up here as well. Eukaryotic cells can have a slime layer. They can have a capsule. But all eukaryotic cells will have typically have some sort of glycocalyx. Our cells, in fact, have glycocalyx associated with them. And what keeps your cells attached to one another, to the layers below them, to create individual organs and things like that, we're talking about our extracellular matrix. So these are components that are non-cellular aspects that usually are made up of proteins and or carbohydrates, things like that, in order to create our extracellular matrix. All right, cell walls. We already mentioned this a little bit. This is going to be the fungi have them. Plants have them, but we're not learning about plants in this class. So the fungi have this. The fungi have it. Oh, and so do algae. I forget about algae. Fungi can have cellulose, just like plants do, or they can have chitin. And that's like what bugs have, in case you didn't know, like on their exoskeleton. So it's kind of crazy to think about, but yeah. So that's a cell wall. You don't really need to know much else about it. It does just the same stuff that it does for the bacteria cells, which is provide rigidity, shape, and stuff like that. Right. Cytoplasmic membrane. We've said that if you're a cell, you have one. All cells have it. Why? Because if you're a cell, you have to hold the stuff in the cell in. And that's what the cytoplasmic membrane does. Remember, cytoplasm is the goo that everything's found in there. So the cytoplasmic membrane is pretty much the exact same, but we are stabilizing our membrane with sterols like cholesterol because we don't have cell walls. So there you go. All right, let's talk about the nucleus. The nucleus is our control center of the cell. That's its main, like big mind picture here. The nucleus is the control center of the cell. Why? Because inside of the nucleus is your DNA. So all of your genetic information always is stored inside of your nucleus. And what is a nucleus? It's just really literally that. It is the area where your genetic information is, and we put a membrane around it. That's what it is. So it's not anything crazier than that. It's where we store your DNA. It's the control center. All that. So what happens is because it contains your genetic material, whenever you are a cell, you don't just have it and it sits there, right? It's not just existing for the sake of existence. It's not only used whenever you're making a new cell. It's used pretty much constantly. Anytime you need a new protein to do anything. Imagine that you ate a new sugar and you don't usually see that sugar, and now you need an enzyme to break that sugar down and use it. Or you need to make this particular amino acid because you've run low and you need to build this protein that needs that amino acid. There's an enzyme for that, or whatever else. Anything that could possibly happen in your cells is run by proteins. Literally all of it. 20,000 genes in your body, the ones that actually code for a product, are going to be related to protein or making the protein. So when I say making the protein, I'm talking about the ribosomes. Ribosomes are the machines that are actually going to be reading basically the many versions of your genes. Well, I want to talk about that in just a second, but they're making the protein. And so if you're making a lot of protein, you need a lot of ribosomes, yeah? So if you need a lot of ribosomes, turns out they are made of something called ribosomal RNA. So if you need a lot of protein and therefore a lot of ribosomes and therefore a lot of ribosomal RNA, then you're going to be making a lot of ribosomal RNA from the genes for that in your DNA. And that happens in an area called the nucleolus. It's not a true structure, it's just an area where we have that going on. All right. I wanted to bring that into context because it's important to understand kind of what's hanging out in the nucleus and why it's hanging out there. We have it segregated in there because, man, you've got 23 pairs of chromosomes in your body. They're all linear. So it's not a big old nice little circle that you can consolidate down. So you don't want them floating off and doing whatever. That's too much for the cell to manage in a big cell. So we have it contained inside of the nucleus. It protects it and it keeps it managed. So it's easy enough to imagine.
Does the DNA like run out as it gets used?
No. So yeah, so good question. So I'm actually gonna go over that. So your DNA is with your cell for the life of the cell. And if you are a cell that is making a new cell, you're gonna make a copy of all of your DNA. Anytime you're doing that, that's called DNA replication. That's one of the first things we're gonna learn about in chapter 6 when we get out of this DNA, okay? So DNA replication is where we make a copy of your DNA for the new cells. But in the meantime, your cell needs to do stuff. So how does that work? So basically, rundown of it. Again, this is all chapter 6 stuff, but I want you guys to have like an understanding of what I'm about to say these things. Like, cool, okay, and you don't know what they're doing and you can't paint the big picture. It's kind of useless. So I'm doing my little workout. Imagine that you have a book that is like the master copy of how to make anything in a cell, right? And that's what your DNA is. Okay, it's the master copy. And the nucleus represents the Smithsonian Institution. Do you go into the Smithsonian Institution and take any aspect of that with you out of it? No, they'd kill you, right? So that DNA stays always in the nucleus. We're not going to take parts of the DNA or anything like that. But let's say I need to make a protein or whatever, making amino acids. I'm running out of it and I need to make my own with the components I have. This enzyme will do that for me. It's encoded in this page of my compendium in my Smithsonian Institution, in my DNA. So what am I going to do? So what I'm going to do is I'm going to take a piece of paper and find that like story or whatever, and then I write down the copy of that one page to make that one enzyme. And that makes sense in practice. So you'll take your copy, you can leave the compendium there, and then you can leave the Smithsonian Institution and take the copy with you. Then that copy is what the ribosomes are going to read. And as they read that copy, translate it into another language, which is the amino acids, is the letters of the language. And then the words of the language, of course, is going to be the proteins. So they're translating into a whole other language. When we first did this, we were looking at our DNA in our nucleus, right? We found that one gene. So we know that's in DNA. That's a nucleic acid. We're making a copy of it. It's in the same language, by the way. So it's a nucleic acid that we're taking with us, that copy. But it's not DNA. It's slightly different. That's a little rickety. It's your handwritten copy. It's what we call messenger RNA. And that messenger RNA is the copy that you'll be actually working with. That's your working copy of that gene. And that is what gets popped into the ribosome, and the ribosome spits out the protein that you want. And that's what you get. That's how that works. So starting in the nucleus, get to your DNA, make your RNA copy, then that's what we call transcribing. You can imagine, because you're writing and writing, so the same language, just DNA to RNA, transcribing. Take it out of your nucleus, and then now we're taking it to the ribosome, and the ribosome is going to translate that into protein, right? So that's the idea of what's going to happen and why, we can say the DNA can hang out there in the nucleus and it's not going to get used up, and it's being kept in its nice little place, and you can make as many copies of whatever gene that you need to and just go visit it. And we do this so much. You're always doing this, like constantly, especially if you think about how many new cells you're going to have to make or at any given time or whatever. So much going on. You're always having to visit that. That is why when we looked at our DNA double helix and everything was held together with just hydrogen bonds, that's why. Because we need to be able to break those weak bonds and get in there all the time. It can't be costing us just energy after energy after energy to really get in there and make our coffee. So now it's starting to come together a little bit. Anyways, so that's the nucleus. Inside of the nucleus, I hate mitosis, you guys. All right. If you do need to make your new cell, you need to make copies of your DNA, but you're also going to have to make copies of all your organelles and everything too. But more importantly, let's talk about what's happening with your DNA when that's happening. It's the only time that your DNA is not going to be in the nucleus. The only time. So you're making a new cell. The first thing you're going to do, your DNA, you're going to make a copy of it. Okay? So it will look something like this. Hey, look at this guy, and it's all like chill. It's spread everywhere out into the open, right? Well, that's how your DNA might normally be. But now we're going to consolidate it down into chromatin and the copies. So if this was your one chromosome that you got from your mother, maternally, anyways, it doesn't have to be whoever raised you, but who, you know, from the mother genes, you have that originally, and you had made a copy of it. I don't know why this is not working the way I want it to. That's not meant to look like K, it's meant to look like an X, by the way. You made a copy of it. So I'm showing you something about these chromosomes that people don't understand. So I'll explain in a second. But if you have your father and you had this chromosome from your father and you make a copy of it, I'm showing you that chromosomes are not X's. I want to make that very clear. They are not X's. And I know every single picture of a chromosome shows them as an X. That's because in science, when we're trying to look at a chromosome, the easiest time to see it is in mitosis, where we've had doubling, we've had copying, we've had everything squished down and consolidated. Your chromosomes are they're only one. Okay. They're just one line. This is what they look like when they're copied. All right. So that's important to understand because we're about to show you what is going to happen with these X's, right? So we have our DNA, we've copied it. Every copy of every single chromosome is attached by the middle to the other chromosome. So we're already nice and equal. This is our like preparation phase. We're getting ready, right? So that we made our copies, we're consolidating, we're getting ready to make a new cell. This is the prophase. So these are the phases, P, mat. This is what Dr. Shearer says, right? He says the puppy pees on the mat, and I like that, so we'll stick with it. Puppy pees on the mat, y'all. So the prophase, if that's a fancy enough word for you, is also where we're doing all of our prep work. Okay, we're just getting ready. All right, the metaphase is the next one. And the metaphase, so this is prophase. This is what prophase looks like. See how we got everything consolidated down, but we're still just getting ready. Let's look at some major changes now. This is metaphase. You see how we've lined up all of our X's in a big row, and it's showing us like 4 X's. All 23 of your chromosomes will line up, right? And they're pairs, actually, so they'll all have copies of 46 or whatever of your chromosomes will be lined up right there with their little copies. And then what's going to happen after that? So this is metaphase. They line up in the middle. I don't know. I didn't use the M, but I didn't. All right. The next step is anaphase. And what's going to happen in anaphase? We're going to tear these things apart. So apart. Right? And then so that's what's going on. So you made your copies so that whenever you tear these apart, now we're going to make our individual cells. This is telophase. So I don't know how you want to word this one. This is a harder one for me. I always say totally done, but you can think totality, totally done, twins, because we're going to have twin cells. But that helps me kind of remember what's happening in each one to use those letters. So, so that's the only ones you need to know is those ones. I don't need you to get in the middle of them, and I don't need to know what the centrioles or the spindle fibers are doing. Not in my class. Okay, that's for somebody else's problem. But that's the idea. So if you have all of your chromosomes, and remember, we have for chromosome 1 and what genes are encoded within chromosome 1, you have one from your mom and one from your dad. We've now duplicated those and they're in X's. And so you will, when you pull those apart, your new cell will have the right number of chromosomes. Yeah. 2 from the mom and 2 from the dad for each of the chromosome pairs. You know what I'm saying? 1 from the mom, 1 from the Jesus Christ. Anyways, yes. Does it make sense though? At least what I'm trying to say.
Yeah.
Yeah. So they'll be in pairs and they'll get pulled apart so that we maintain the right number of chromosomes in the daughter cells. Because that's it. Because once we split that up, that's the cell. All right. So that is how mitosis works. This is when we're making copies of our actual like body cells. You cut your finger, you can get new cells to heal it up, and that's mitosis. You're a little tiny baby and you're growing into an adult. That's mitosis. All right. All these cells have the exact same genetic information. We're making copies. It's just based on their environment, what genes are going to be turned on, and that'll kind of determine like what cells they'll become and all of that. We're talking about stem cells becoming tissue and all that. So that's the idea. Meiosis now. I'm not going to draw out meiosis because you don't need to know that in this class, because I'm definitely not going to be talking about, you know, sex ed in here. Okay. So meiosis is the generation of the gametes. You can imagine that this would be a problem. So I'm going to actually kind of draw this out in the board, just make more sense. For mitosis, so I'll just put mito for this one. Actually, I'll put cis because we're going to talk about mitochondria later. I don't want to get super confused. Mitosis, you are going to go from 1 chromosome. This is like from your mom, and 1 of that same chromosome from the father. You can call it exosome number 1. And they do have them like numbered like that, right? The pairs and whatever. So this is your exosome number 1, chromosome number 1, your mother and your father. When you're going through mitosis, these guys are going to become, for the mother, an X, and for the father, an X, right? Because we're doubling them, so that whenever we pull them apart, right, we have new cells that have the correct number of chromosomes like we just started out with in our original cell. So that's how mitosis works, all right? So that's great. So we have the right number. So if we had only 1 chromosome here, we've got only 1 chromosome pair here. We've got 1 chromosome pair here and 1 chromosome pair here. We're all happy. We're the same, all of that, right? But meiosis is quite a different concept, because if you have that same chromosome and you had 1 from the mother and 1 from the father, then that's going to become the X's, whatever. And then that's going to become, let's say, even divide. Let's not even put them into separate souls yet. Let's just say 2 from mother, 2 from father. When they get pulled apart, yes, they'll be in separate cells. But at this point, if they were in the same cells like these were, and then this were a sperm, and that were an egg, you guys see where the problem is? Because if this is a sperm and this is an egg, and the sperm has the full chromosomes, the 46, and this has the full chromosomes, the 46, whenever these come together, that's way too many chromosomes. What you need is just 1 of chromosome 1, not both copies, just 1. So in meiosis, basically, if we had, let's do it a little bit more. If we had these as our product, it's simple enough to imagine that, but these can't come together like if this was a sperm and this was an egg. So what happens next is literally you'll have 1 with the mother version, 1 with the father version, and then the same for the other ones. And so you get 4 each with 1 of that chromosome 1. Not 1 pair, but 1. So you might think this doesn't have very much genetic variation or anything like that, and so how do people get to be so different, and why don't you look exactly like your sister or something like that? Well, that's because whenever you go through all this process, it's actually a lot more complicated than this. Of course it is, because science, right? But you'll have overlapping between the mother and the father chromosomes, and you'll have switching of the actual genetic information on those chromosomes. Isn't that just, that's actually happening whenever you make your gametes. Now, for women, they've already got theirs made, comes with the package. For men, they'll be generating their cells, and then they'll need to be able to do this quite frequently. But That's meiosis. So that way, whenever you have an egg and sperm randomly come together, they would be able to make a proper number of chromosomes. Okay, that's meiosis. Really all you need to understand about that. I don't need to go into too much detail. If you really want me to talk about it more, ask me later, because that I feel like is one of those topics that can get really complicated. All right, so we have our nucleus. It has all of our info in it, but our DNA never leaves the nucleus. All we ever do is make a copy of that note and take that out to be read by the ribosomes. So what happens when that goes on? So you made your RNA, your messenger RNA, on your little note, your working copy. You're going to take it out to ribosomes that are found on the surface of the endoplasmic reticulum. The endoplasmic reticulum basically encompasses the nucleus, because that's what the nucleus does, is it, sends out the notes for that to be made into protein. That's pretty much what it's for. So the endoplasmic reticulum is this like nice, like wavy, many layers going on, and it's studded with ribosomes, and those ribosomes will make your protein there. And then the proteins that are made in there can be modified with lipids or whatever you want to put on it, whatever it needs to be functional, right? So that's pretty much what it does, and it also can make those lipids and stuff. So this is where the making of things goes on, the endoplasmic reticulum. The rough ER specifically is where we're making proteins. It looks rough because see those little blue dots? Those are our ribosomes that are hanging out on the surface of the endoplasmic reticulum. It's a membrane that the ribosomes are hanging out on. It's a membrane. These are all membrane-bound organelles. Ribosomes are not membrane-bound organelles. They're just ribosomal RNA that'll clamp down onto the messenger RNA. It shows a little picture of that. We'll get into more detail of how that works in chapter 6. Next, we have the Golgi apparatus. So you're at a factory, let's say. Somebody's ordering a whole bunch of, air fryers. And so there's a factory that's making the air fryers, and that would be your ribosomes in the endoplasmic reticulum. Okay, so we're making the air fryers, we're putting the right polish on them, we're making sure they're the right color people are ordering, and then they're just going to sit there and wait because they need to be sent to the warehouse at that point to get packaged up. So that in the warehouse, they'll get packaged up and sent to whoever ordered them to the right address and whatever, right? That's the Golgi apparatus. The Golgi apparatus is our transport facility. It's going to do the packaging of what you made, and it's going to transport to where it needs to go. So it makes a lot of vesicles. So membrane-bound vesicles. All right, so this, let's go. Cool, that's good. Here's a little picture of what they look like. They look quite similar to one another. I'm not going to make you guys pick mountain of pictures. What do we say? Yeah, so it is sort of like an assembly line where we have the nucleus crapping out some mRNA that's going to go through the endoplasmic reticulum to make a protein that then can be sent to the Golgi apparatus and actually packaged and put wherever it's supposed to be. Outside of the cell, inside of the cell, whatever. You guys ever seen those little videos of like the little walking protein guy that's like carrying the huge, massive vesicles, right? So anytime that you need to move a vesicle anywhere in a cell, that happens every time. So anytime you're making anything at all and you need to move a vesicle, it's going to be one of those little protein guys walking along, carrying that giant vesicle of whatever is in there to wherever it needs to go. Imagine how much energy it takes to do all of these things. Quite a lot of energy. So the energy to make the guy walk, the energy to have made the guy, because he had to get made first, right? All of that stuff, making the vesicle in the Golgi, like these things don't just happen spontaneously, right? Man, getting real complicated. That's because the cells are. Right, next are the vesicles themselves. The two kinds that are kind of more important, I guess you could say, are like the vacuoles. We'll start with that because they're boring. They're storage vesicles. Plants have giant vacuoles that are filled with water, and it's why if you don't water your plant, it'll start to wilt and just kind of like collapse down, because the vacuoles actually help it maintain its structure and all that. So if you don't water it, it'll just wilt. So just storage. But the lysosomes, okay? So zomes, anytime you guys ever hear this in this course, zome, that is talking about vesicles, right? So lysosome, I guess I did ribosomes, those are not vesicles, so just whatever, forget it. I tried. The other ones tend to be, okay, please. So the lysosomes, these guys are vesicles that contain digestive enzymes, including lysozyme, which we kind of had on our little quiz a minute ago. Lysozyme is an enzyme that we can make in our cells that breaks down peptidoglycan. So your cells can just make it. And typically we find it in our cells in areas we'll see it in our tears, and we'll see it in our sweat and in our saliva. All of that area will have lysozyme in it, getting ready to break down some peptidoglycan that it just might encounter. So it's like our first line of defense, right? But our cells, remember our phagocytes that eat up the bad guys, they come up to the bacteria and they engulf it. Well, they put it into its own little vesicle, its own little package. It's like kind of like a cell. Think of it as like a waiting cell at like the jail or something, right? So you're in your own cell and you're just waiting, you don't know what's going to happen. That's your phagosome, all right? Because the phagocyte ate it up, now it's a phagosome. Then you're going to take your lysosome and containing all of the digestive chemicals that it has, including lysozyme, including toxic oxygen products, including acids, things like that. You're just going to merge it right in, and that's going to be what eats up your bad guys that you ate up. So that's how that stuff works in your cell. That's how important the lysosomes can be in, basically breaking down bad guy stuff. All right, mitochondria. Hopefully most of you guys know what the mitochondria is, but it is the energy generator. It's the energy factory of the cell. It makes our energy. That's the big guy concept, okay? They have, right, so we already said we were gonna get into how this is like bacteria, right? But they have two membranes, the outer membrane that just surrounds the whole thing, and then on the inside, a membrane that's all folded up and has just stuff going on all along it to increase the surface area. And those are the cristae, that's the folds. And the enzymes that are on the cristae, those proteins that are dotted along it, those are involved in aerobic respiration. And that's where we get our ATP. So your mitochondria are correlated quite closely with how much energy you're making. If you were a sprinter, you're taking, like, if I were to take a sample from a sprinter's quad, and then I take a sample of my quad, and you look at our cells next to each other, their cells individually, a sprinter will have so many more mitochondria just in their cells. Yeah, they'll have bigger muscles and more muscle cells too, but in their individual cells, they'll have more mitochondria as well. Why? Because they always need more energy. They need to be ready at any time to sprint and deal with that. What happens when a sprinter runs around, the track? They aren't going to get sore afterwards, but I would. Like, I sit on my butt all the time. So why does that happen to me? Because I have only so many mitochondria to make only so much energy. And once I use that up and I need more immediately, what does my body do? It does something very akin to what we would call fermentation.
Lactic acid.
And it makes lactic acid as the byproduct. And that's why you get sore muscles, because you didn't have enough mitochondria to make your energy. And so you had to do something else to survive. So that's what happens. And that can happen for other reasons as well. But that's the idea. So you can, if you work out more and more, you get more muscle cells and you get more mitochondria as well, so that you don't have soreness anymore when you've been working out regularly. And that's why that works the way that it does. So it's kind of cool to think about. And so you have the folds, and then inside of that fold, that little cavity that's kind of in there, it's called the matrix. It has its own ribosomes. It doesn't, no other organelle has its own ribosomes. Everybody else is using the cell's ribosomes, not the mitochondria. The mitochondria has its own ribosomes. It has its own DNA in a circle, one chromosome in a circle. We've got two membranes, right? We've got our own DNA, we've got our own ribosomes. As it turns out, the ribosomes that are in your mitochondria resemble bacterial ribosomes, not ours. They are noticeably different. So there's that. So already crazy stuff that indicates that perhaps your mitochondria used to be bacteria. They also divide on their own. If your cell is dividing, it'll get signals that it needs to divide, but it doesn't get controlled by the rest of the cell like the other organelles will. It will divide on its own, and it divides itself by a process known as binary fission, which is what bacteria do. Okay? And it will divide itself. You do not have any genes in your entire DNA at all that build a mitochondria. Your endoplasmic reticulum, you've got genes for that. Your Golgi, you've got genes for that. Building a membrane around your nucleus, there's genes that control all of these things, but not your mitochondria. So that's interesting, right? So how do you get your mitochondria? So you have the mom and the dad and the sperm and the egg come together. So where do the mitochondria come from? All all that. Does anybody know? What's from the mom? That's why the egg is bigger. One of the reasons why the egg is so much bigger than the sperm. It has to come with mitochondria. And that's where all your mitochondria come from. Everybody's mitochondria comes from the mother side of their DNA. And this is the truth. And so tell me that's not wildly interesting. At least, yeah. And so you can actually, in a way, trace back almost in a way, looking at the genes and telling you what they can tell you about it. But the DNA of your actual mitochondria all the way back like, through like organisms that existed well before us and that sort of stuff, because they had to come from our precursor cell back in like the literal like way before prehistoric life existed, like the first cells we're talking about here. So that's crazy to think about. We can't go back and test those, obviously, but that's the concept of where it comes from and why we all have so much similarity in those sorts of things when we look back at it. So really cool.
Do they develop that way because that's so important? Like they need to not be reliant on the
why the mitochondria are
right,
separate
why
from.
do they have their own DNA? Why do they? Because they don't.
That part is probably what an evolutionary biologist would probably have a lot more answers. But the real fact of the matter is, it's just because they haven't needed to change that,
Okay,
right? So there's been no selective pressure on that. Yeah, they've been working out okay so far, right? So there's a picture of your mitochondria and what it looks like. You can see that inner membrane and how it's all folded up and all that. The chloroplasts are very, very similar. Obviously, their structure is a little bit shifted, but those little stacks that we have going on, those are the inner membrane, if you will, of their mess of what they have going on. And so all of that is going to be, they have their double membrane, they've got the crack going on inside of their cells that has chlorophyll. The chlorophyll gets excited by sunlight. And when that does that, it like triggers this whole cascade of reaction to help them make a little bit of ATP. And what they do with that little bit of ATP isn't just survive. They don't just use that. It's way cooler than that. And we're not going to talk about it now. Okay. So this is where I say that plants should rule the world. Because they got something way cooler going on with what they do with their ATP from photosynthesis. Okay, don't worry, we'll get to it in chapter 10. I know you're excited. Okay, so our ribosomes make our proteins. I really don't need to go into a whole lot of detail about this, do I? They have them separated in size. We have, as eukaryotes, and I'm not going to test you over this just so you know I'm not, but we have 80S. That's the size of our ribosomes, and the bacterial ones are 70S. So they actually are pretty different. And this is a measure of sizes. And yeah, these do not add to that because that's just how the measurements, it's not measured by adding, it's measured by putting it into an ultracentrifuge and seeing where it sits in a gradient. And where it sits in a gradient determines what size you are. And these are called Svedberg units. So whatever. But I'm not gonna test you on it. If you're interested, there it is. And I was telling the other class, I was like, I'm terrified of an ultracentrifuge. If you've ever seen centrifuges, they'd be like, a little box sitting on a table and you can put all these tubes in and they'll spin and everything will settle at the bottom. An ultracentrifuge is like one the size of like a washing machine. And it's like, it'll only still hold like 4 tubes in it, but it can hold, it'll spin so fast. It's like disastrously fast if it wasn't balanced correctly. Right. And so, of course you learn how to use it properly and you learn how to balance it. And it has like little triggers that's unbalanced, just like your washing machine does probably and all that. But yeah, so they terrify me and no, that's not for me. All right, so the cytoskeleton is what kind of sounds like it's your cell's skeleton. It gives your cells shape as well as something for your little guys to walk along. So this is what your guys would use as a pathway to carry your vesicles around, would be these guys here. I'm not going to get too much into that. Yay, our first questions. Always seem to get like so caught up in talking about the nucleus that I can't catch up with other stuff. All right, let's run through these real quick, guys. Do your best guesses, all right? I ain't gonna hold it against you. All right, what is the biggest difference between eukaryotic cells and bacterial cells, David?
Bacterial cell walls, bacterial cells all have cell walls, but eukaryotic cells some have cell
That's a good one. That's one you remembered, and that it's factual, right? We have some bacteria that don't have the cell wall, like those mycoplasmas. There are exceptions, but that's a good one. So the difference I'm looking for here, remember, don't overthink this. The eukaryotic cells and the bacterial cells, the eukaryotic cells have a nucleus. Yeah. So don't overthink it. That's what I mean. All right. Next question. But that you weren't wrong. I'm giving you the point because you made a good argument for it. All righty. That was which one? We did this one. Yes. Okay. All right. The flagella, bacterial flagella versus eukaryotic flagella. Can you tell me at least one difference, Brittany?
Oh yeah. They're 10 times thicker.
Yeah.
Complex.
Yeah. So thicker. They, the eukaryotic ones move like a whip, right? All right. The eukaryotic nucleus. So what is its big guy idea, purpose, Taylor? Yeah, that's it. We're getting now. We're getting there. All right, and what is the function of the rough ER? This is Christy. Yes, it is. Good job. Yes. So yeah, it's rough because of those ribosomes, and that's what the ribosomes do is crank out the protein, right? All right, here's one that shouldn't be too bad. The mitochondria, what does it do, Emily?
It makes our energy.
Yep, that'll work. All right, guys, I think that's it for that part, and we'll just crank through these next portions. Yeah. That's probably the only questions we're gonna have. Yeah, it's probably the only question we're gonna have to do. We have like 15 minutes or something left. So let's do the fungi, okay? Fungi. Yes, we do have mushrooms and all that stuff that you can see, but we're gonna talk about the microscopic ones, the molds and the yeasts. Yes, yeast falls into fungi, all right? When you look at a fungi, if you're looking at a mold, okay, a proper mold, and you have a piece of bread and it's literally growing mold on it, and it's fuzzy, big old lump, let's say. The little hairs, if you were to look real close, those little hairs that come off, those are the hyphae. Okay, that's what that term is about. I'll tell you the terms that you need to know for this. Definitely know the difference between hyphae and mycelium, which we're going to get to in a moment, okay? Fungi, they get their nutrition from, like we do, heterotrophs, so just breaking down stuff in the environment normally, or they can break down things that are dead and decaying. Those are saprobes to do that, or they can be parasites. What kind of fungi are parasites? Like athlete's foot, for example, ringworm, that stuff. Causing harm to us. Okay, the mycelium. I don't care about septa, and we're going to talk about spores in a moment. But the mycelium is like you're looking at that piece of bread, the little hairs were the hyphae, for the big picture of the bread, that whole body of it, that's the mycelium. What else do we care about? We'll talk about spores in a minute. So if we need to reproduce as fungi, we would necessarily be either breaking our piece off, like have a piece break off and then go into another piece of bread that's nearby, or we can just keep spreading where we are, or we can reproduce. That can be sexual or asexual. I am not going to talk about sexual reproduction with the fungi, okay? We are only going to talk about the asexual, right? As it turns out, the asexual reproduction involves something called spores. There's nothing to do with the endospores, okay? So that's why I have that written up there, because I want to make it clear that they are not related to one another. Okay, so the spores here are an asexual reproduction thing for the fungi. Now spore, and what are these guys for? Reproducing. So for reproducing, is that in the rough times? Right, no, it's not. So it's going to be in the good times. You can see how these are not related. Yes, I understand that that says spore and that says spore, but just, I don't know why science did that to you, but that's how we got it, okay?
So to remember the difference in rough times, the ending,
yes.
Spore without the end, good times.
Good times, yes. We're not having an ending, right? That's one way to look at it. Also, if you guys just think about mold and mold spores in the air. If I say it like that, you're like, oh yeah, mold spores. That's when they're reproducing. That makes also a little bit more relation to what's really going on here and how you can separate these. So hang on to that idea of mold spores. You know what they are. So then you know that bacteria and endospores are not like the fungi spores, and you know that they're that kind of stuff. All right. So So it has two forms of spores that it could make. So it's you're one or the other as a species. So one species does that kind, but other species might do the other kind. That's what I'm trying to say. So for example, penicillium that we get penicillin from, or aspergillus, it's like one of the common molds in our environment, they do conidiospores. Conidiospores come from this structure called a conidiophore. I will not make you know the difference between the two. Okay, I want you to know what conidio versus whatever the sporangio is. Okay, when you see them. So conidiospores, they are the open ones, and then the sporangiospores are the ones that are contained. They'll have a cover, and the sporangiospores, they will just burst out of that cover when they're ready to do their thing. But these guys are just always, the conidiospores are always open. I don't have a way for remembering this that's good, so I hope that you guys can find something that works for you, because the only things I ever think of are wrong. Like I was the opposite, and it's not helpful for anybody. So yeah, don't ask me for that one. You guys can figure something out that works for you. Good job. So they do make sex spores as well, but we don't need to know that. Yeah, you can use this information and those structures and look at them under a microscope, like they look like that, to identify them. So it's pretty identifiable. You can see that's the sporangiospore one and the conidiophore one. You can just see that already without being in a picture, like drawing. It's real. You do have diseases that are caused by fungi. Obviously, you can be allergic to fungi, and being allergic to fungi will create a allergic reaction, that inflammation in your sinus cavities, let's say, and that will make you more susceptible to having an infection from the fungi as well. So people who are allergic to mold are also more likely to have mold-based sinus infections, for example.
So if you're allergic to mold, you can't have penicillin or,
You can, yeah, it won't be related to that. It shouldn't be anyways. Yeah, so, but yeah, so there's, let's see what else, and opportunistic infections, of course. If you have a lowered immune system, you're going to be more susceptible to some of the more basic molds that shouldn't be really getting anybody else any problems. Or the, if you ever know anybody that's been on like Humira or something like that, that has like a lowered immune system, they're more likely to have candida infections, so yeast infections on their body, because their body can't keep the yeast that's on their body normally under control. So that's an opportunistic. It took the opportunity.
so like when we send urine and they look at it under a microscope, if it had yeast in there, they could actually see it in
Yeah.
the microscope, like
Yep.
whatever the pictures look like.
Yes, they could. Presuming that there was enough of it in there, right? Okay. So, right, we also know that we can use fungi. You can use maybe the fungi itself to make things like penicillin or whatever, but you can also use yeast for making bread or what, beer or whatever else. Let's get into the Protista. Protista is the kingdom, then it has subkingdoms, algae and protozoa. Okay, subkingdom algae and subkingdom protozoa. These guys are single cell organisms, and the algae are like part of one of those important things that makes oxygen in our earth. So the algae, the plankton that falls into the algae category, those are the ones that make our oxygen. We kind of know that. You guys know about how the plankton and the algae does that stuff, but we maybe not appreciate at the scale that it does it, but now you guys know. For the protozoa, now this is what we're more concerned about as far as disease goes, because algae isn't going to cause itself disease. It might produce a toxin occasionally with like red tides or something like that, but that's not what we're interested in. The protozoa can actually cause infections, and you've probably learned about more of them than you think that you have. Okay, so they are single cell. I want to be very clear about that. These are single cell organisms, but their cells and their organelles have been kind of reorganized, so they look like mouths and digestive systems just in one cell. That's kind of crazy to think about. And they have what looks like legs, but they're just one cell. So crazy. And they can live all over the place. Waterborne is usually what we would think of the most. So they can get around. There's 4 classifications of the protozoa. The first classification, let's say, is the ones that don't move, the non-locomotive ones, okay? Then there are these other 3. We learned about the flagella, what they do. We learned about cilia and what they do. Then the pseudopods. Pseudo is like fake, it's not quite real, right? Pseudopods, think of podiatrist, so fake feet. So what they do is, these are the amoebas. So think of amoebas, you guys know that. They goop out and then pull themselves along with their little goopiness. That is an amoeba. They fall into pseudopods. And here's some pictures of these guys. Cool. All right. Let's do this. the trophozoite and the cyst. Let me get the flag. Tie this in there. Where did I put it? Y'all, I can be trusted with a marker. Here it is. All right. Cyst. The cysts for the protozoa are dormant, resistant. You see me looking over at this other one, you know where I'm going with this. But let me try to get it in the right order. And then these guys are made in rough times. Sound familiar?
They're the endospores of
Yep, the cysts are. That's why I have these up here so you guys can see, because I don't want you to get them flip-flopped in all this. So the endospore and the cyst. Do you guys remember what the active form of the bacteria are? What it's called? If you don't. Vegetative. Put a little line under this so you kind of know this little stuff thing. Vegetative, these are the metabolically active ones. Don't get confused by this fanciness. I'm calling it metabolically active. It's just ones that are actually doing shit. Right? They're doing stuff. These guys are just hunkered down, waiting it out. These are doing stuff. So if you see a bacteria doing anything, that's what we're talking about. So metabolically active here. And then over here, they also would have to have a metabolically active one, right? You can't always just be this. So if you're going to be active in reproducing and things like that, their version, obviously you can see on the board, is trophozoite. I know it's a fancy word. This is metabolically active. Okay, so that's a crazy word. Trough. You guys know what a trough is? Like what pigs eat out of. It's really what I'm talking about, like actual trough. Okay, if a pig is eating out of a trough, is it dormant? No, right? So trophozoites, trough, these are the guys that are metabolically active. Okay, so that's why the cyst has to be dormant. If I were to say even like this, something is encysting, or you could even say if somebody had a cyst on their body, is it like going around, moving around on its own? It's not growing some bacteria, but you know what I mean? It's not itself going somewhere like you are. So that's something to think about. So there's that. So you have your words up there to not get confused about, you know, it's kind of difficult for some people. And we can, of course, by looking at them, separate them by how they look as well. Okay. Parasitology is the study of protozoa as well as the helminths. Okay, and I'm going to run through the helminths real quick, but it's not going to take very long at all. So real quick, the protozoa, Naegleria and Acanthamoeba, these are our brain-eating amoebas. If we're talking about Giardia, you already heard me say about that. You heard me say about Trichomonas. We also have Trypanosoma and Leishmania, which aren't very common here, so you probably might not be familiar with them. Here we have Plasmodium, probably one that you should be aware of, even though we don't have a lot of this. And I want to say a lot of this, we don't have a lot of this, but we do have it, y'all, malaria in the United States, okay? Wow, how? You might be saying, we don't have malaria in the United States. We absolutely do. Go to Florida and tell me. Go get bit by a mosquito there, y'all. So we do. So it's not common to see it, but some of the mosquitoes carry it now. So just, yeah, bad. So malaria, toxoplasma. Anybody know toxoplasmosis or heard of it? Anybody heard of that? It's a protozoa found in cats naturally. That's their normal place to be, and in cat poop and in their litter. So if you are pregnant and you're told not to handle cat litter, that is why, is because the disease caused by toxoplasmosis. It's a flu-like illness for people that are just normal and healthy. Otherwise, if you are pregnant, it might not cause you too much harm, but it can be abortive. So that is serious. So don't handle the cat litter. Make sure anybody you know that's pregnant, make sure they understand that this is not a joke. Okay, the last group is the helminths. And there's one word up here that you need to know that I don't have on the slide, and I literally always forget to put it in the slide. The flatworms have their own little word, and it falls into helminths, which is platyhelminthes. Again, I'm not going to make you write it out. You just have to be able to recognize it. Platy, flat, sounds like flat to me. It's the only one that's going to sound like flat, right? So there you go. Flatworms are platyhelminthes. You can divide the flatworms up into the cestodes, which are the tapeworms, and the trematodes, which are the flukes. Those are flatworms. We also can have roundworms, and our roundworms are called nematodes. They don't have any breakdown words. They're just the nematodes, okay? So those are our fancy words, platyhelminthes, cestodes, and trematodes, and then the nematodes for the roundworms. Okay. These guys cause terrible diseases. They're bad. I don't know what to tell you. They have actual organs. They are multicellular. These are animals, literally. These are kingdom Animalia here. So again, the adults are big and you can usually see them, but it's the larvae and stuff that you can't see. And that's usually the part that you're going to get infected by if you're drinking contaminated water or eating contaminated foods or whatever. So yes, they have very complicated life cycles that may involve having to go through certain kinds of hosts in order to get to how to grow up or whatever. It's crazy complicated. And then the host that they actually reproduce in is called the definitive host. That's where the adults reproduce. Okay, how do they infect you? If you were going to get infected from your food, soil, water, or infected animals that you may have eaten, like undercooked pork, for example, you can do it by oral intake. So eating undercooked pork is one example, but drinking contaminated water would be another example. And penetration of unbroken skin. Let that help you guys sleep at night. So now I'm talking completely about like you go to swim in one of these countries that has some of these diseases, and it has been contaminated by one of the flukes, for example, and they are in the water, then it can go literally through your skin into your body. That's the schistosomes that do that. There's other ones that can go through the bottoms of your feet. That's the hookworms and the whipworms. So yeah, these are mostly, of course, in third world countries. That's why we don't have a whole lot of knowledge about them here in the United States, because in the United States, we generally don't really look too much towards the third world countries. Tell me I'm wrong. So yes, we do have some of these in the United States, of course. Humans can have pinworms, which is like usually little kids, you see this, right? They're playing in like a sandbox or something, and then a couple days later they got an itchy booty, and then you take your kid to the doctor and they're like, put a piece of tape on your kid's butthole and look at it the next morning, and if there's worms on it, then it's pinworm. This is real.
Not in the middle of the night. They wait till the middle of the night to come out. So your kid wakes up freaking out. I took mine to the ER. I had no idea what pinworms were. They just laughed their head off at me.
Yeah. They're like, most people don't. Most people think it's super like this crazy weird thing is going on. It's super common actually.
I really get the meds for this at Walgreens.
Yeah, it's over-the-counter medication. Yeah.
Like I know what pinworms are.
Yeah.
Like it's a common thing.
Yeah,
in
animals. Yeah, they carry it. That's where usually the kids will get it from, is from playing outside and contact. Anyways, now you guys know, and it is not uncommon here in the United States. So much so that you can literally go to Walgreens or Walmart or whatever, and there's over-the-counter medication for that specifically. So
What is it, Jim?
it's just called pinworms.
Where does it come out?
Your butthole. Your butthole.
What is it? Is it like cream or something?
No, it's like pills that you take. So
It's liquid, actually.
is it liquid?
It has to be liquid to coat it.
to coat it,
And
there you go.
It's really gross, and you have to do it exactly 24 hours later because of the cycle of
The cycle of their life and the larvae
whatever.
production. Yeah.
And it's banana flavored. I don't know why. It's awful. And you're supposed to treat everyone in your house if one person has it because it's so easily transmissible and you can't see the eggs and what.
It's like if you get lice, but for your butthole.
Right.
That sounds exactly the same, right?
Fall out of the butthole.
Yeah.
In the middle of the night, yeah. In the middle of the night. Yeah. And it's very itchy because whatever they lay their eggs in is itchy. So
Yeah, something. Kids are, yeah. If they're playing outside a lot, so just, I would say, encourage your kids not to put their hands in their mouth without washing their hands, as much as you can control that.
Or don't have to.
Yeah.
This is the best birth control like ever.
I know. Other ones, obviously, we could talk about like what we do with animals. We're talking about heartworms. Did I tell you guys about my dogs? I think I did. Yes, meatball and spaghetti. And they had to get treated for heartworms and stayed in their little kennel for like a long time. And it was sad, but they should be good now, I think. But no, I don't know about meatball, you guys. Listen, let me tell you about meatball really quick. Meatball. So I let him go outside. He's a chunky boy. All right. He's real chunky. And I don't really understand why he's so chunky, because I give him less food than I give to Spaghetti. And Spaghetti is a smaller dog. And he's like, we call him a noodle because he's spaghetti. He's a little spaghetti noodle. He's a little skinny guy. So he gets less food. I'm always like, Meatball, why are you so chunky? Like, I don't know what to do for you, buddy. Well, he will go outside and eat all of the bugs. All of them. I caught him one time. I went out into the backyard and he had a cicada in his mouth. He's like a chihuahua mix. Like, he's like not the size of a chihuahua, but like a little bit bigger than that. He had a cicada in his mouth, and it's like all in his mouth, like actually. And so he's like sitting there with it in his mouth. I'm like, no, Meatball, put it down. And nope, he's just like, like trying to get it down before I can get over to him. So I think I found out why he's a chunky boy. But I'm also worried about, like, if you eat like bugs like that, that's where dogs can get like more parasites and stuff from. So hopefully he doesn't get, he has to go get his checkup appointment soon. But yeah. There you go. It could be anything, honestly, because yeah. So we're not going to do the questions. Like I said, we'll just be done here. But yeah, these diseases are all over the world. We do have some in the United States. They're not just in third world countries. So
at least your dog actually eats the cicada, does just stand there.
Right. I don't know. It's kind of traumatizing because the like wings are all sticking out of his mouth and it's like buzzing while he's chewing on it. And Meatball. He's got the right name, y'all.